Hello everyone, welcome back to the cell signaling course. If you can recollect, in the last class we were discussing about a very unique type of receptor tyrosine kinase signaling, where it was a contact-mediated signaling, which meant that receptor and ligand both were present on the cell surface. We will continue with the cell contact-mediated signaling. However, what we are going to see in the class today is that receptor does not have any integral enzymatic activity, unlike what we saw for EPH-effrin signaling, which was a contact-mediated signaling, and receptor had the tyrosine kinase activity. One unique feature of the EPH-effrin signaling was that it was a bidirectional signaling. It activated repulsal signaling, and it had many more additional features and was found to be regulating a lot of boundary formation in tissues as well as early embryonic development. So when we talk about the other signaling, which is also called Notch-delta signaling, this is similar to what we have seen for EPH-effrin, where both receptor and the ligand, they are membrane bound. Here Notch serves as a receptor and delta serves as a ligand. And then the neighboring cells, which are part of a tissue, which are arranged in the juxtaposed manner, they engage in this contact-mediated signaling. Both of them, whether it is Notch or the delta, they lack any enzymatic activity. So they basically undergo a lot of changes. Notch, particularly as a receptor, undergoes a lot of changes to induce a transcriptional regulation. And these both do not associate with anything which has the enzymatic activity, like we saw in case of JAK-STAT pathway. Receptor did not have the enzymatic activity. However, it associated with JAK kinases which had the tyrosine kinase activity. Here in Notch-delta signaling, neither the receptor or ligand, or they do not associate with anything which has the enzymatic activity. So we will see how this Notch-delta signaling works, but it is very important for many developmental events which happen in the organism. So what are the components of the Notch-delta signaling? As you would expect, you would need a receptor-like molecule and you will need a ligand-like molecule. Since both are membrane bound, both of them have the extracellular domain. And through these extracellular domain, they engage in contact-mediated signaling. So if you imagine a scenario where there is one cell which is expressing the ligand, which in this case is delta, and the other cell which is expressing the receptor, which is Notch here. So basically the cell which is expressing delta is called signal producing cell. And the cell which is expressing Notch is called signal receiving cell. If you recollect, I have been telling that in these signaling, the receptor expressing cell is the one which is doing signaling, which means that changes happen inside the cell which expresses the receptor. And an exception of that we saw in the previous case of EPH-effrin, where both, whether it is receptor expressing or the ligand expressing cells, were doing the signaling. So what happens in this case, that you have the two components, one which is the receptor and the other one which is your ligand. They engage in this interaction and then signaling goes on in the receptor bearing cell. We will see some similarity between Notch-delta and EPH-effrin signaling, which we saw in previous classes. So here again, since both of them are membrane bound, signaling will continue into this region. Unlike EPH-effrin, here receptor is not undergoing phosphorylation. No phosphorylation. Rather, it undergoes a proteolytic cleavage. Remember the proteolytic cleavage we saw in case of EPH afferent signaling, where afferent bearing cell was undergoing receptor was undergoing afferent cleavage, which allowed the internalization of or disengagement of the signaling. So here the proteolytic cleavage at this particular site releases a component which is called NICD, notch intracellular domain, and this domain has the sequences which direct it inside the nucleus, where it regulates the transcription process. So generally you will notice that this signaling is actually affecting the process of development and formation of organ systems and so on. And we will see also that how this signaling is important for regulating the movement across the cells and instructing other cell type what kind of signaling should go on, what signaling should be happening and so on. So normally what will happen, that you have this, suppose this receptor, this is activated, which means that there should be a cleavage here, there will be additional cleavage here, which will ensure that this NICD is translocated inside the nucleus, where it will work on genes which are under control of these set of molecule, which generally is kept off. Under normal condition, the components here which are regulating the expression of genes, they are put into a repressor mode. Which means that when no signaling is happening, the NICD is intact with the receptor. The complex which is present here in the 5' UTR region of target genes, they are switched on into a repressor mode. That's why there will be no transcription. But when the ligand binds to it from the other cell type, the signaling is activated and NICD is generated. This NICD will move inside the nucleus, bind to this complex which was repressor type, and the binding of NICD to repressor complex relieves the repression. Basically, that's when it activates transcription. So if you think about the components, there are two components here, one which is receptor, which is notch. Multiple types of notch are found if you think about the mammals. So notch 1 to 4, and then you have the ligand which is seen as delta, and there are other ligands which have been discovered and they are called jagged. So together the combination of different notch and different delta jagged things actually induce the signaling. And basically what happens inside, the complexes which are serving as a inhibitory or repressor in the beginning, which turns into an activation mode, they are also present over there, and some of them are like CBF and RBPL kind of molecule. So basically you have two components, receptor and ligand, and then there is a machinery which is inside the nucleus which is on into the repressor mode, but signaling and the availability of an NICD into the nucleus turns it on into a active mode. Now, if we simply look how this signaling moves forward, I gave you a brief detail. So, generally notch is produced through ER Golgi route and it is directed to the plasma membrane. And here in the Golgi, there is a processing which happens which cleaves the extracellular or the domain which is inside the cytosol, and then the reassembly of this entire structure happens and you form a structure like this where receptor is moved to the plasma membrane. So, basically before notch as a receptor moves to the plasma membrane, from the Golgi network it is processed by proteolytic cleavage and this molecule is now put onto the membrane, plasma membrane. This is, I would say, this is your receiver cell and this is your producer cell with respect to the ligand. So, the ligand which is here as a delta, they will come into contact and activate the signaling. Remember, similar situation was there with EPH efferent signaling. When signaling had to terminate or active signaling was actually leading to the separation or repulsion of the two cells, and for that you needed some sort of cleavage. Okay. So, same thing will be happening here where this productive engagement of the signaling between delta and notch induces the cleavage. The first cleavage happens here. If I can say this is the first cleavage, then it is the second cleavage. What this will do? This will remove the notch extracellular domain and it will be taken back into this producer cell. Then third cleavage which happens inside the cell in the intracellular region, which generates this notch intracellular domain. There is a set of molecule which work here, they are called gamma secretase. This is the protease which is working and generating the NICD. And as I told you, this NICD will go inside the nucleus through nuclear pores, where you have this complex, repressor complex. And NICD binds to that and turns on the transcription. So, basically notch delta signaling is working by converting the repressor property of the molecule which is bound to the chromatin into an activator type and thus induce the signaling. So, what happens further inside the notch delta signaling and what are the consequences of notch delta signaling? So, what is understood from several research and different experiments which are performed in organisms or the mutant analysis which has been done suggests that they are important for organ system development like hematopoiesis, vasculogenesis and neurogenesis. If you recollect in EPH efferent signaling, we saw how EPH efferent signaling was important for vein, artery demarcation. In case of plexus as well as the vessels which will from a common precursor vessel, they were becoming veins and artery. This notch delta signaling plays very important role in neurogenesis. Basically, the conversion of an epithelial tissue into a neural tissue is regulated by notch delta. We saw how EPH efferent signaling was contributing in ensuring that neurons find their end, make contacts and establish, mature further all those spine regions and the dendrite regions of the neuron. So, let us see how notch delta signaling is working in neurogenesis. We all understand that during embryonic development, once you have the gastrulation happened, which generates the 3 germ layer. So, from the germ layer which is called ectoderm, the one which is present apparently outside and this ectoderm, a part of ectoderm is defined to become a neural ectoderm, and this neural ectoderm undergoes the signaling and converts into the neural tube and further brain and other CNS component. And inside the brain or central nervous system, we understand that neurons are one type of cell and there are other type of cells which are called glial cells. So how the differentiation of neuron type of cell and glial type of cell happens? So you think about epithelium where you have multiple cells. A scenario projected here that there is one cell which is in the center and these are cells which are in periphery. So during early developmental event, all the cells which are shown here in this picture, whether it is, if I consider this as cell 1 and then 2, 3, 4, 5, 6, 7. So there is one cell which is in the center and 6 cells are in the periphery. All these cells which are in this region, they both express notch and delta. So basically signaling goes on in all direction. Each cell is signaling to the other cell. And this is why this epithelium remains undifferentiated. But as the development happens, progression of embryonic development goes on, there is change in the expression level and the cells which are on the periphery, they start to express notch, more of notch in peripheral cell. And the cell in center switches to express more of the delta. And from what we saw in the previous slide, the cell which is expressing the notch or the receptor will undergo the signaling. So the message which is given to the peripheral cell is that the fate for the central cell has been identified. In this case, the central cell which expresses more of delta acquires the fate of neurons. So eventually what will happen? You think about the scenario. Now this becomes a neural cell or neuronal cell and this will become non-neuronal or glial cell. So notch delta signaling is ensuring that how the tissue is getting organized or differentiated in becoming one cell type versus the other. So generally what I showed you and what goes on here is that basically you have a scenario, what I was meaning over there when you have neighboring cells, and one thing which is shown by notch delta is lateral inhibition. Which basically means that each cell is inhibiting the other cell by virtue of notch delta signaling. I am just marking some of the inhibitory signal, but you remember that it is bidirectional because both cells, which is central cell and the peripheral cell, are expressing notch and delta. As the time progresses, you now get to a situation where productive signaling is going only in one direction, which is basically happening in the peripheral cell. So here you have now unidirectional signaling as the development is happening. So earlier the cells were unspecified because no commitment was done, because every cell was receiving the inhibitory signals. However, in this case, as the development progresses, only the peripheral cell receive the inhibitory signal and the fate of central cell is defined. So it has specified its function. So if you consider the scenario of if I say the central cell becomes neuronal cell, so eventually from here what will happen? You will have a neuronal cell if I can just write N as for neuronal cell and G for glial cell. Right? So normally this is how differentiation will happen. But if for any reason the switching happens or one of them is not working, so if suppose this neuronal signal fate is not there, then all cells become glial cell. Or if the other way around goes wrong, all cells become the neuronal cells. So this bidirectional signaling in the beginning and then switch into unidirectional signaling is very important for such fate coming through the Notch-delta signaling. Another example which is very clearly demonstrates Notch-delta working is the production of socket and shaft cells in mechanosensory bristles of fruit fly. So fruit fly head region has a lot of these bristles which have mechanosensory function, from which they sense multiple movement which are around them, and the basic structure of that cell is that you have one socket cell, and from here you have a shaft which is emerging out, and on these shaft you have sensory molecules which help it perform the mechanosensory function. So these cells actually come from a common precursor cell. So this precursor cell will divide asymmetrically to give rise to two cells. Remember, this cell is expressing both Notch and delta. If I say N and D, both are expressed. However, what happens is that there is another molecule which regulates the Notch function is localized to one side of this cell. So when asymmetric division happens, asymmetric only in the sense that this molecule which is inhibitory to Notch is present on one side or going in only one cell. So you will have one cell which receives the inhibitor and the other cell is perfectly fine. So you will be seeing that the inhibitor receiving cell, this Notch signaling is off, and the one which is not having the receiver inhibitor will be showing Notch signaling on. The cells which actually show Notch signaling off, they become shaft or sheath cells. But the one which have Notch signaling on, they become the socket cell. So what you understood that the signaling going on in one cell makes it a socket cell, and the one where signaling does not go on becomes the sheath or shaft cell, right? And that is by virtue of presence of this inhibitory molecule only in one of the cells. So what will happen if suppose for any reason the inhibitor is absent or inhibitor has been negated? So both cells which are coming out of this division, they will becoming the socket cell because inhibitor is absent. And for any reason, if this distribution, if suppose now the cell for whatever reason it had the inhibitor like this and it underwent division in this way. So the two cells which are present, they both will become the cell where Notch is absent. When Notch is absent, it will become the sheath cell. So how you saw that basically Notch-delta signaling is ensuring that there is one cell's fate specification which goes on. Also basically what I told you over there, that inhibitor going into both cell becomes the sheath cells, inhibitor inactivated makes both as a socket cell, right? So presence of one molecule versus other, it becomes very important. One more example which comes from the C. elegans, where this Notch-delta signaling is inducing mitotic versus meiotic fate choices. So if we think about the gonads in C. elegans, so there are cells, and there is one unique cell which is present here, which has tentacles. It's called distal tip cell. This distal tip cell has these tentacles which is expressing the ligand, and there are these precursor cells which are in this region, and they basically express the notch. So as long as these cells are in contact with the ligand, notch expressing cell and the ligand delta expression, this induces the signaling in them, and they do not undergo meiosis. They divide mitotically. Because in this early region you want cells, more precursor cells, so they don't need to undergo meiosis, rather they will undergo mitosis to increase the population of cells which will be giving rise to the more differentiated cells. When cells divide and move further away from this distal tip cell tentacles, no more productive notch delta signaling can take place. What is happening here? That these cells are, although expressing notch, but they cannot engage in the signaling. Now this is where fate choice happens, the switch happens, and they start to divide meiotically and produce haploid gametes. So you notice that how signaling is directing a cell type to divide either mitotically or meiotically. So you can assume if this, for any reason, if this distal tip cell is not present, then from the very beginning you will see meiosis going on. Or for any reason, if your ligand is not expressing in these distal tip cell, you will see that again it is starting to divide meiotically from very beginning. So what exactly we have learnt in today's class is that C. elegans or Drosophila or for any organism, this notch delta plays very important role. And in general, when we talk about the signaling, there is a signaling which must discontinue. In previous case of EPH-Ephrin, you saw how the digestion was ensuring that signaling discontinues, or the entire membrane region was taken up by transcytosis. So what happens here in case of notch delta, if you consider this cell and this cell, this is expressing the notch, and this is the one which is expressing the delta. So the region which is in proximity to the membrane in the extracellular space, this is where a domain arrangement is there which is normally in a coiled state which is inhibited. Inhibited in the sense that it cannot undergo proteolytic cleavage. So when the signaling starts and push and pull happens, it will eventually make a situation where this region, coiled region, is more opened up, and you will see that notch, this is now amenable to the cleavage by another set of proteases which are ADAM proteases. And because of this notch extracellular domain being released, the delta bearing cell will take this entire thing up in this cell. and thus it will discontinue the signaling. So importance of this extracellular domain and its conformation is very big when it comes to continuing or discontinuing the signaling. So in general, what we have learnt in today's class is that Notch-delta presents another example of cell-to-cell contact-mediated signaling. The receptor here lacks enzymatic activity, and it also does not associate with any enzymatic activity. Receptor is activated, or signaling is activated in this sense, I will say, by cleavage of the receptor. We saw the cleavage happening in two different ways. One which was extracellular, which was helping removal of this complex from the cell surface or from extracellular matrix, and the other cleavage which was happening intracellularly, which was generating the notch intracellular domain, which went inside the nucleus and activated transcription. Then this we have seen example that how this signaling is important for neural epithelium specification. It was important for Drosophila mechanosensory bristle, basically socket or sheath cell identification. And we saw example how this Notch-delta signaling plays very critical role in mitotic versus meiotic division choices in C. elegans, where the signaling activated to ensure or ensured that mitotic division is preferred. So we saw some parallels between EPH-afferent signaling and Notch-delta signaling, and something which is very unique for Notch-delta signaling with receptors lacking any enzymatic activity, receptors not associating with any enzymatic activity, rather they are carrying out signaling by transcribing a set of gene and turning the system which is inside the cell from a repressed state to an activated state. Thank you.